Hi, this is Gwen and Chico. I am doing my presentation today on aspirin and prostaglandins, and we're going to look at the pathways and how inhibition of the prostaglandins occurs. So this is my presentation on aspirin. The first thing I'd like to talk about is how do we get aspirin anyway, and it's acetylation is the process of how we produce aspirin. We have salicylic acid, and we use an acetoxy group, which is this thing with the oxygens, and under a very strong acid, and this is phosphoric acid, and it catalyzes the reaction to form aspirin, which is also called acetylsalicylic acid. Acetylation is the process where we take a hydrogen from this hydroxyl group, and the part of this acetyloxy attaches to there, and this is the structure that we get that forms aspirin. The pathway of aspirin acts as an acetylating agent, as we described, taking hydrogens from taking an active hydrogen from a hydroxyl group and attaching an acetyl group, and is covalently attached to a serine group in the active site of the COX-2 enzyme, which I will be discussing a little further in the presentation. Mechanism of actions of aspirin. The primary one is acetylation and inhibitory effect on platelet aggregation. So aspirin is very good for reduction of inflammation. It's an analgesic. It also prevention of clotting, and is a reduction of fever. So acetylation is related to decreased production of prostaglandins and TXA2, which is thromboxane A2. And these are from irreversible inhibition of the COX enzyme, which I will explain on the next paper. Other mechanism of actions that aspirin can do is an uncoupled oxidative phosphorylation in the liver and the cartilage in mitochondria. And what that does is aspirin buffers and transports hydrogen ions competing with the ATP synthase. Another mechanism of action is that modulates signaling through this NF-K beta, which is a transcription factor complex that plays a central role in biological processes, including inflammation. Aspirin also induces formation of NO, nitrous oxide radicals in the body, and that is to reducing inflammation by reducing leukocyte adhesions on the cells. So aspirin has many mechanisms of actions, but primarily we're going to focus on the acetylation effect of aspirin and inflammation, and how it affects prostaglandins. Prostaglandins are not from a gland. They're actually at the cell membrane, and what happens is when you have an injury to a tissue or you have an infection that causes when there is a fever, you have pain, you have an increase in temperature. This section I'm going to talk about the prostaglandins, and when we have a fever, we have pain or inflammation, anything like that where there's an injury, the prostaglandins are related to the inflammatory response that is stimulated by the immune cells. And prostaglandins are signal messenger molecules, and they are not formed from a gland. They are actually stimulated at the site of the injury or of the tissue or the infection area. So the normal pathway of how prostaglandins work at that cell membrane, it starts by deriving from fatty acids in the cell membrane. and they use an enzyme called phospholipase A2, and that cuts off the fatty acids in the membrane, and they're catalyzed next by cyclooxygenase, and that is catalyzed by adding oxygen, and a ring is formed. So the fatty acid that is seen in the cell membrane to start the process of prostaglandins is this arachidonic acid. It's derived from the cell membrane. Here we see the phospholipase A2 that will derive it down into this. This is the structure of arachidonic acid, and then it goes to the cyclooxygenase, where that enzyme forms, the oxygens are added, and then here we're starting to form the ring. What happens from here is that we end up developing an intermediate prostaglandin H2, which is this guy. And then further enzymes break down the prostaglandins into different molecules. This is prostaglandin F2 alpha. This is the thromboxane A2, and this is prostaglandin E2. Prostaglandins also function in blood clotting. They constrict or dilate blood vessels, and they produce protective mucus in the stomach. And they all come from this cyclooxygenase time. I drew out this a little bit better. This shows the normal pathway to prostaglandins. That's the outside of the cell, that's the inside. Here's the fatty acid in here, the arachidonic. So if the fatty acid comes through, it gets catalyzed by the phospholipase A2, creates the arachidonic acid, as you see. And then now we have the cyclooxygenase COX pathway, which comes down here. And there's two isoforms that come from that. You have a COX-1 isoform and a COX-2 isoform enzyme. This COX-1 functions more in the body. It's always present in the body. And like I mentioned, there's a protective coating in the stomach, protective coating of mucus in the stomach, and that's that prostaglandin. And then the thromboxane is platelet aggregation. It helps with forming those blood clots. So this guy is always present in doses. But this one, the COX-2 isoform enzyme, is induced. It's an induced prostaglandin when we see that inflammation, fever, and pain response from the cells. Aspirin works by inhibiting non-selectively the COX-1 and 2 enzymes. So when that happens, there's no prostaglandins that will be produced, leading to reduced pain, fever, and inflammation. So when these are blocked by aspirin, then the prostaglandins are not made, and now we have a reduction in the pain, fever, and inflammation. Aspirin actually prefers to inhibit more of the COX-1 versus the COX-2 enzyme, and aspirin acylates the serine amino acid, the serine 530 amino acid residue present in both so it actually aspirin acylates the serine amino acid residue in both COX-1 and COX-2. Specifically over here, I wrote out the mechanism. As you can see, this is salicylic acid, and it forms our aspirin. This is aspirin, and under COX-1 enzyme, we can see that the serine residue 530 is active with the OH, but as we go here, we're going to inactivate it, and that's where the aspirin actually blocks it and comes down. It makes it inactive, inhibits the prostaglandins, and inhibits the synthesis. And so at that position, exactly on the serine 530, is where the aspirin will block that active site. On this side, the aspirin is here, and the COX-2 inhibitor, we see that it actually chooses a different type of pathway. The serine 16 hydroxy, that serine residue, it